All right. Since we have a packed agenda, I think we're going to go ahead and get started. So again, thank you all for joining us today. Just a reminder that we have our YouTube channel, which you can access here, which features all of our previous webinars. And if you're interested for any feedback, questions, would like to be added to our listserv, please email us at help@cfdeknowledge.org. I'm going to go through some announcements and events, and then we'll go ahead and have our featured speakers. So first up, our upcoming 2026 webinar schedule. Today we'll hear from Illuminating the Druggable Genome, and upcoming in June will be CM for AI or Cell Maps for AI. We're excited to have this upcoming schedule, and if you are available and would like to present November 20th, we are still looking for a speaker, so please contact me or help at cfdeknowledge.org. So we do have this slot open. And with that, we also have a couple upcoming conferences that we'll be attending. Next week, which is June 3rd and 4th, will be the Festival of Genomics, Biodata, and AI in Boston. I will touch upon this in a second, a little bit more in depth, but we'll also have a wide CFDE presence at the ISMB conference, or Intelligent Systems for Molecular Biology, which is July 12th through 16th. So if you're going to be attending either of these conferences, we'd love to have you stop by, talk to us, and learn a little bit more about CFDE. As I mentioned, we're going to be at the Festival of Genomics next week. We have booth 1A in the exhibit hall. This will be across from the Genome Dome. If you're interested in learning about new updates, personal demonstrations for these resources, you can visit us there. We also have a talk, which will be June 3rd from 4:10 to 5:40 p.m. in room 50, which is From Prompt to Provenance, AI-enhanced contextual workflow across the Common Fund data ecosystem. And we have speakers from the NIH, Biomarker KB, and the Knowledge Center. We're excited to have this session, especially since the Festival of Genomics is geared particularly around AI this year. And so we're hoping to give an example of, how we can guide researchers and using knowledge graphs to basically bridge together data sets and more. And so we're really excited to be presenting there for this time. So again, if you're going to be at the Festival of Genomics, please feel free to stop by our booth and our talk. Earlier this month, the Training Center held their 7th annual BioIT Fair Data Hackathon. These are just a couple of the pictures from the events. We had such a wonderful time participating, and we really got to see, CFDE in real time with these hackathon attendees. So thank you so much to the Training Center for hosting this event and for having us participate. And then just a reminder, if you want to learn even more about CFDE, the latest Decoding the Data Ecosystem podcast has been released earlier last week. And so this podcast, as a reminder, it's for anyone who's passionate about potential omics research, or addressing, health challenges and things like that. So whether you're a seasoned researcher or a student, this is for you, as there's going to be various insights from the programs within CFDE and beyond. Just a reminder, there is a funding opportunity pilot projects enhancing utility and usage of Common Fund datasets, which is closing June 23rd. And so the idea of this opportunity is that there are many valuable and widely available data sets that are generated from the Common Fund programs. And the idea is this could demonstrate and enhance the utility of selected Common Fund data sets. And the award recipients are asked to provide feedback on the utility of Common Fund resources. So as a reminder, this is closing June 23rd. So if you scan the QR code, that will take you to the opportunity itself. One last event that we have upcoming is the Incubator for Pan-NIH Resources. And this is designed to be July 9th and 10th at the Neuroscience Center. And we are going to be using this session as an opportunity for collaboration, shared themes, and generate pilot projects as a way to explore scientific interoperability. And one way we're planning on doing that is through the scientific themes, looking at gene sets as embeddings, knowledge graphs as the central integral layer between data sets, and AI-enabled connections to bridge the gap between the systems. So we're really excited about this event. There's going to be a keynote speaker, panels, breakouts, and a great opportunity for us to move forward for the scientific interoperability. So if you're interested in attending, please scan the QR code at the top here. We are very excited to meet with all of you in July. So now I'm going to hand it over to Illuminating the Druggable Genome team, who will be presenting on their data sets and tools. So with that, I will stop sharing and we'll hand it over to the team.
Thank you, Mackenzie. Let me see if I can get my screen share going. Does that look all right?
Sorry, I'm on mute. Yes.
Yeah, okay. We from the IDG program are pleased to participate in this webinar of the CFDE public webinar series organized by the CFDE Knowledge Center. Thanks to the Knowledge Center and other CFDE leaders for inviting us to present IDG news and updates today. I am Jeremy Yang, co-PI of the IDG DCC Data Coordinating Center. My fellow co-PI being Christoph Lambert. I'm a research associate professor in the Translational Informatics Division of the Department of Internal Medicine at UNM. Dr. Lambert is professor and chief of the division and also lead of the informatics core of the CTSC, Clinical and Translational Sciences Center at UNM. Also presenting with me today are Jessica Main, staff scientist at the NIH National Center for Advancing Translational Sciences, NCATS, and FAIR-ified 2.0 lead developer, and Praveen Kumar, research assistant professor in the Translational Informatics Division, and KG2ML lead developer. So those will be their topics. Incidentally, or maybe not so incidentally, Jessica earned her PhD at UNM and was a postdoc in our division before joining NIH. And thanks to the CFD public webinar series, and thanks Mackenzie Brandis for this nice announcement. So couldn't resist sticking that in our slide deck. This webinar has three parts for us three co-presenters. First, I'll present an introduction and overview with history and news. Then Jessica Main will present updates to the IDG knowledge portal, FAIR-ified. Then Praveen Kumar will present KG2ML, an ongoing project and platform for discovery driven by harmonized common fund datasets. Then I'll conclude with a brief wrap-up. This presentation will, to some extent, introduce the IDG program and its data resources with relevance to CFDE scientific use cases. However, there are so many more IDG resources of potential value, but they are readily findable and accessible via an extensive corpus of documentation and publication. So kind of a tip of the iceberg situation, I would say. So anyway, please feel free to ask questions via the chat or via any other channel. And we are, as Mackenzie said, to try to stick to our time limits, asking you to hold off on the oral questions until after we get through our presentation. Okay. The IDG program was designed to improve our understanding of understudied proteins from the most drug-targeted protein families. 1, G protein coupled receptors, GPCRs, 2, protein kinases, and 3, ion channels. 2, catalyze research in understudied genes that have high potential to improve clinical therapeutics and human health. The NIH Common Fund sponsored IDG from 2014 to 2023, full 10-year span, supporting a diverse array of experimental and computational research and producing a rich variety of data sets. DruggableGenome.net is the IDG website, and pharos.nih.gov is the data portal, integrating many other data resources from IDG, Common Fund, NIH-wide, and other sources, global community sources. Bottom left from druggablegenome.net, that graphic illustrates the extensive IDG network of institutions and collaborators spanning the US and including European collaborators. Bottom right is from an IDG Tool Fest event with a sampling of logos from IDG Discovery Tools and data resources. Again, a kind of a sampling. Though the main awards supporting IDG have ended, the program has aged rather gracefully, I would say, with partial thanks to CFDE, but also thanks to the continued interest of the scientific community and the continued importance of the central goal and need for new drug targets. Aging gracefully means not doing everything you used to do, but focusing on sustaining the most important and valuable activities. So I can't resist saying that at my biological age, I can kind of identify with IDG in this respect. Publication and citation counts provide a clear measure of the importance and impact of IDG and trends over time, though through the present. For example, in the last 2 years, since the last webinar, public CFDE webinar, by the way, since June 2024, from June 2024 to now, May 2026, citations have increased from 51,000 to 79,000, up 55% in the last couple of years. So things are continuing, you would say. I want to share some of the most recent IDG publications to my knowledge. The breadth of the IDG consortium was such that many of the experimental and computational research activities have continued in some form, but may or may not be explicitly associated with IDG. So most likely I'm missing some, sorry to those, which are either fractionally or largely IDG-driven papers. The publication featured here is a book chapter authored by the FAIR and Target Central Resource Database TCRD development team at NCATS and UNM, and includes NIH Science Officer Ajay Pillai of NHGRI. I'm mentioning him because he really played a key role in conceiving and guiding IDG throughout its lifespan. It was good to see him as he stopped in for a visit at the spring meeting in March to see how things are going with CFDE. So I know he continues to have a great interest in a lot of informatics programs at NIH. These are publications from the last couple of years, 2025, 26. The featured paper is from the Ave Mayan lab at Mount Sinai about harmonisome, which integrates over 100 sources, well over 100, a resource developed for IDG and a key component of TCRD and FAIR-ified, distinctive in many ways, but among them, its ML readiness. Things have basically been harmonized into essentially a giant table ready for machine learning processing. The Mayan lab has continued to maintain, update, and enhance harmonisome, which includes a database, web UI, API, and maybe now, naturally, an LLM-powered chatbot. So check it out if you're interested. These are papers I've already listed, but with a bit more to say about them due to their ongoing efforts. The TINX update paper was published in 2024 with postdoctoral fellow Vince Metzger at UNM as the lead author and project manager. TINX has been a key resource for IDG since its initial release in 2016 and a key component of FAIR-ified. TINX stands for Target Importance and Novelty Explorer. It's a bibliometric method which identifies novel co-occurrence associations between diseases and genes from the PubMed-based text mining it's from Lars Juel Jensen's lab in Copenhagen, Denmark, or it has been, and that text mining workflow, which is also used in the well-known diseases resource and others. Recently, in the past few months, data scientist Jack Ringer has been refactoring and updating the TINX technology stack for reliability and efficient hosting. That's not done, but that should really make it more supportable and more performant as well. TIC-TAC, the next paper, means Target Illumination Clinical Trials Analytics with Cheminformatics. And this paper was one result of the thesis of Jeremiah Abbock, who graduated from UNM with his PhD in chemistry and chemical biology, December 2024. He's now a scientist at LabCorp, but has continued as an UNM affiliate and contributor, collaborating with student research assistant Vivek Sharma Panthi to produce a new API and web UI. So that work continues. And the third paper is the new KG2ML paper, so I'll leave that to Dr. Kumar to describe in his portion of the webinar. More news updates and work in progress. Manjul Pradhan is a computer science PhD student with us, and GRA, who joined our group in the fall of 2025, last fall, and has already contributed a great deal, including developing and updating APIs for key IDG resources, particularly Drug Central and FAIR-ified. Accordance with REST and DRS standards, supports interoperability with a variety of consumers and agents, but a focus has been on the CFDE Cloud Workspace Galaxy platform in cooperation with CFDE Cloud Workspace Implementation Center, QUIC, I guess we call it. Interoperability with Galaxy and development of example workflows is an important and effective channel for outreach and training. For example, via the recent, this month, Galaxy Training Academy. Most of you know how global that community is. Likewise, developing workflows for the Data Resource Center Playbook Workflow Builder platform is a great way to reach another community of users. More news and something that's pretty exciting for us. Earlier this month, the Drug Central team committed to a plan for an update this year, which has been delayed for a variety of reasons, generally good reasons, and changes affecting supporting resources and personnel. Throughout that time, we've recognized the importance of maintaining, updating, and further developing this highly used community knowledge base, which is an open access compendium, we call it, of approved drugs. They're high confidence targets and has rigorous semantics, identifiers, and cheminformatics. Dr. Tudor Preya, now Chief AI Drug Discovery Officer of Dompe Pharmaceuticals, Italian-based firm, and Emeritus Professor at UNM. Still, he conceived and led the creation and development of Drug Central over a decade ago. Earlier, two key contributors were Oleg Ursu, who's now a principal scientist at Vertex, and Jamie Holmes, who's now been promoted to an IT administrator in our main campus at UNM. So those two are not available, but most of the team is back with with key contributor, research professor Christian Beloga, and also an important new contributor, the talented research assistant Vivek Sharma Panthi. So we have a good team in place. Another key contributor is Dr. Soren Avram, professor at the Coriolan Drăgulescu Institute of Chemistry, trying to say that right, in Timisoara, Romania, and others there, including Dr. Ramona Kirpan, who was a former UNM postdoc that we all miss. Already, the Romanian team has curated many or most of the new drugs and targets since the previous version in 2023, and the technology stack has been reconfigured and migrated from DigitalOcean to AWS by Vivek. He's also begun work on an LLM-powered, I'll say MCP-ready, retrieval-oriented generative RAG chatbot. So we're pretty excited about that. Other enhancements are in development, meaning new code and components are ready for integration. For example, many RD-kit-based cheminformatics functionalities. This is a screenshot of a recent Google Analytics report of Drug Central usage, indicating 115,000 active users and some other statistics. So it's just here as an indicator of the continued importance and value of Drug Central, and really a motivator for us regarding its community of users. As with previous version updates, we also expect an increase in usage following the upcoming release and publication. And my final slide before handing it off to Jessica Main is about our training and outreach activities from IDG. Recently, we participated as Mackenzie mentioned, the BioIT World Fair Data Hackathon organized by the Training Center. And our project was conceived by Tudor Preya, illuminating the molecular basis of drug side effects. Tudor and Manjul Pradhan were in Boston at the hackathon to lead the project team. So that went well, just a few days ago. Next, our 2026 summer internship program has begun, which features CFDE datasets and IDG use cases, and is closely based on last summer's CFDE-sponsored internship program, which really was a pleasure for everyone, I would say, and we consider very successful, and is described in this poster on the right of the slide, which Vince Metzger and I presented at the UNM Health Sciences Education Day. So that's available online as well if you're interested. So I think that's it for me. Hopefully I'm roughly on schedule, and I'm going to click through slides for Jessica Main. And so I hope that's her first slide.
Yes. Yeah, thanks, Jeremy. Go ahead and well, actually, I kind of assumed that this group might already know what Pharos is about. And then I kind of realized as you were presenting, maybe I should have had it more about page, but you did a good job explaining it. It comes from the IDG program, obviously, and developed. But NCATS, I guess I have to disclose then, the NCATS team has taken over the hosting and maintaining and optimizing the Pharos 2.0, which we're hopefully releasing within the next couple of months. But I'm going to present to you what the differences and the changes that are being updated from Pharos. But we are calling it Pharos 2.0. So go ahead and next slide, please. So just a little high-level overview of what Pharos is. It's the back end of what all the IDG, all those resources, all those databases that came from IDG. We kind of harmonized and created this huge interface that the data sets and those annotations from those resources all come together, making some drug targetable insights or disease associations and whatnot. The biggest thing that what I always advertise though for Pharos and a lot of users that use Pharos is the TDLs, which are the target development levels. And that's what we categorize these proteins at a different level. So T-dark is we don't really know anything about them. T-bio, there's a little bit of biological evidence, like some literature or some Go annotations that are associated with them. And then you get bumped up to T-chem, and there's some small molecule activities or some kind of experimental data that's out there. And then lastly, T-clin is what we categorize when there's an FDA-approved drug. And I'm very excited to hear that Drug Central is going to be updated and maintained with all that new resources, because we heavily use Drug Central to categorize these T-clins. I just put a little thing in there about who uses TDLs on more of an open source, and not just like for researchers, but we also Unipro, if you go to their links of their Unipro IDs, proteins and human proteins, there's a little tag that says what the TDL is, and the same thing for PubChem and Kimball. Translator, we do have the TDLs ingested into that program, but it's a little under the back end still, hasn't into there because they're still creating their whole message interface as well. All right, next slide, please. So previously in the current GUI right now, the graphical user interface, it's still in the current this amount of proteins. So the targets that are now in there right now are 20,412. And then you could see on the left that the separate categories of T-bio, T-dark, T-chem. But so clicking that again to show the next. So the enhancements, the updates, though, now we have about 240 more proteins. And I'm going to explain a little bit later why that jump is there, but also because we've updated some resources, creating this whole semi-automatic pipeline that automatically updates from the resources that we're pulling in. Go ahead, next slide. So just to have a visual of the TDL counts, illuminating the druggable genome, right? So you'd imagine the more information, all the more data that's being submitted into the world of research, you would imagine that the T-darks are going to go down, right? So luckily that's what happened. So 5,501 now is only 5,000. The T-bios, the reason why those went down is because if you see in the T-chem, there's a significant increase in T-chem. So the T-bios went down, but the T-chems increased. And that's also because there's a lot of more ligand information and activity data sets out there. And we have had an increase in T-cleanse, about 30 hits on that one. Next slide, please. So as far as the user interface, nothing's going to change on that front. It's going to stay the same. The facets and all the things and annotations that you see currently on that website is not going to change. Next slide. And that also means that the GraphQL, the FAIR API, is not going to change as well. It's going to stay the same. Next slide. But we are going to have a new tool. So originally, the backend was called TCRD, which is a relational database. We still have that MySQL database that's pulling from the graph, the GUI, but we're also creating a knowledge graph that's going to be kind of used as a tool in essence. And we'll be able to have we're still deciding on how we're going to advertise that or let users play around with it. It's still up for debate because the graph is growing pretty big right now, and we got to figure out cloud, all that stuff, all those fun things that you got to, troubleshoot through. But we will have that option available sooner than later. Next slide, please. And also, this is what I'm most proud about because this is my part of this. We now include isoforms. So before it was just canonical proteins, and now what we're doing is we're using the whole UniProt human protein list, and so that includes not only just reviewed but also unreviewed, and that's where kind of the some of the isoforms come from. But if you've ever used UniProt, they actually have this algorithm where they assign isoforms within their set. So it's like a UniProt identifier, and then they'll have like a -1 or a -2 that indicates the isoforms. And so we follow that pipeline as well. But we're also, if there's no indication from UniProt, we're also harmonized with Ensemble and a few other different resources. And so there's like this filtering process of what we define as an isoform versus just an alternative product. And we also keep that tag of if it's unreviewed from UniProt to be aware of that. But so from this graph, though, this new Pharos graph that we're created, the TDL algorithm is staying the same. So like I said before, TCLIN, if there's if there's a drug that's approved for it, it's TCLIN and then so forth. So TChem, TBio, TDARC. Next slide, please. So now that since we are including the isoforms and the unreviewed proteins, now that to those TDLs list jump significantly, right? So it's like almost into the 200,000 proteins now. But you'll notice that the TCLINs now are 1841, which you're like, wow, that's amazing. But no, that's not what that means. It's still the 732. What that means, though, is the isoforms that fall under those canonicals are also getting assigned that TCLIN category. And then it's kind of the same reasoning through the rest of TChem and TBio. There is a little bit difference in the TBio and TDARC because of the algorithm and what the filters and rules that we use to calculate those. The TBio is because there's an ensemble hit from the Jensen PubChem score, which gets a little hairy on that front. But it is interesting to still see that some of the TBios are now TDARCs, the isoforms are obviously, you would imagine that the TDARCs are mostly in those because they're understudied, right? Because there's not little published on them. Next slide, please. We also now from that graph, we have granularity now instead of just like it's called, they always, as a biologist originally, whenever I see gene and they interchange the gene and protein, but they're meaning the same thing, I finally said, no, I want to separate them. And so now there's gene, transcript, and protein level. So now there's more granularity on the targets. Pharos does at the high level, it's protein, but then now from the graph, you can actually go back and track to the different transcripts to even to the gene. And at the SNP level or the variant level, and obviously I just presented on the isoform level. And what this does is now you can kind of have some insights, and if there's a SNP that's associated with the disease, that'll be embedded in that graph, or if there's like a transcript that's a different variant that's associated with the disease. And also, I was hoping to find some more insights in what my direction I'm going in is to see if there's any specific isoforms that drugs hit for the proteins, but for drug discovery aspects. But anyways, next slide, please. And from all this, so NCATS is very highly involved in the translator initiative. I didn't put too much description on what that is because it is very complex and very lots to describe, so I'm not going to do that. But I hope with this group, I think you might be familiar with what Translator Initiative is about. They also created this BioLink data model, and that's what we are following as well. So the Pharaoh's graph is directly compliant with BioLink data model. And what that means is the controlled vocabulary. So they call it categories and predicates, but if you imagine they're nodes and edges. And it's basically a naming system, so controlled vocabularies, and we follow that ontology pipeline. So then later in when we go to ingest Pharaohs into translator, it'll be easily interoperable with their KGX pipelines and ingest pipelines. I think next slide. I don't know if I have one. Maybe not. Oh, okay. So on that front, though, in a kind of it is feeding into Pharaohs 2.0, but NCATS IFX group, we are actually creating this whole harmonization, BioLink translator compliancy pipelines that are because we have different tools within our group too, like Cure ID, which is a rare disease patient input, and so it's all messy free text. And so we have this resolving normalization and standardization pipelines that all harmonize and to be compliant with BioLink and translator ingest. And so this also feeds directly into the Pharaohs 2.0 graph, all these different open sources that translators are already pulling in, and we harmonize with our data sets, and then that's how we make it BioLink compliant. I think that's it.
Thanks, Jessica. And
yep.
I'll just quickly mention, as I did, that during a recent CFD ontology working group meeting, BioLink was brought up and the importance of consistency or interoperability, rather. So I would say that is very good to know and rather harmonious.
Yes. Yep.
Okay. I think we're ready for Praveen Kumar. Praveen, you ready to take it away, please?
Yeah, thank you, Jessica and Jeremy, and good afternoon, everyone. So in next few slides, I'm going to talk about our published work, Knowledge Graph through Machine Learning. And in this work, what we did, we integrated Knowledge Graph and positive unlabeled learning. Next slide, please. So in this work, we developed a Python-based pipeline to discover some unknown association between biomedical entities. So the knowledge graph is based on the knowledge we have, like in literature or some well-known database. But there might be some association between some entities which have not yet been discovered. That's why we used this pipeline and developed this pipeline to discover those unknown associations. And there are three key components of this KG2ML pipeline. One is a condensed knowledge graph that we created using the TDKG from Jonathan and team from the University of Pittsburgh. This paper is available on bioRxiv on DDKG paper. And the second component is our Pulsar paper. Christoph and I developed this algorithm, which uses positive and unlabeled learning. And this has two components. One is based on selected completely at random assumption, and SNAR is selected not at random assumption. And third component is also our work from IDG team. Jessica was the lead author of that paper. And we are the way we select the features is inspired by this protein graph ML work. Next slide, please. So this is the complete pipeline of our KG2ML work. So what we do using this DDKG or UBKG database, which is very giant, containing data from like several DCCs, we create a condensed knowledge graph. It contains nodes and edges that we wanted to have for our research. And we run SQL, Neo4j SQL to identify positive genes and unlabeled genes. Since our KG2ML paper was based on identification of genes which might be associated with given disease. So that's why in this plot, in this figure, I'm showing genes, positive genes and unlabeled genes. And using those positive and unlabeled genes, we identify all the features. And once we have features and labels, we apply this Pulsar algorithm to get the alpha estimate, which is like what fraction of positive is there in the unlabeled set. And this Pulsar PU learning method also returns the calibrated probability for all unknown genes. Next slide, please. So this is the complete pipeline, how we create the condensed knowledge graph. We take the whole original DDKG and then run some Python script to identify nodes and edges that we wanted to have in our condensed knowledge graph. And using those nodes and edges, we create a dump file, and then we load that dump file to the Neo4j. And that's how we have smaller subgraph based on the DDKG. The reason why we created condensed knowledge graph, because we know DDKG has lots of information data from like several DCCs. So we didn't want all the information in our KG2ML pipeline or KG2ML work. So this condensed knowledge graph gave us better interpretability, and if you run the similar query to get the information from DDKG, it takes lots of minutes. But in case of condensed knowledge graph, it takes few seconds to get the results. Next slide, please. So here is an example to show why we did not use the DDKG in its original form. So you see, when we tried this SQL to get information for chronic heart failure, so we have 1,077 nodes and 13,150 edges. And the same query, when we executed on condensed knowledge graph, we got 215 and 460 edges. So you can see it's difficult to interpret all the information from DDKG. And here it's very clear. So that's the reason why we decided to have this condensed knowledge graph. And I would say our condensed knowledge graph work was inspired by KG work from Avimayan group's work. They previously did this subgraph thing. So we got inspiration from that work and created this condensed knowledge graph. Next slide, please. Okay, so now the question arises, why we did not use the supervised learning. So the associations which are not known, maybe no one has studied yet, so we cannot call them negative. So for the supervised learning, we need to have both positive and negative labeled examples. But since unknown association may be either positive or negative, we don't know the answer. So that's why we decided not to use supervised learning and go with pure learning. So in pure learning, we have labeled positive examples and unlabeled examples, which comprises both positive and negative examples. We don't know which genes are positive and which genes are negative in the unlabeled set. So the goal of this pure learning is, okay, identify what fraction of the unlabeled is positive and assign potential for the potential positive and negative among the unlabeled examples. So our Pulsar method gives both things, like proportion of positives among the unlabeled examples, and it also assigns the calibrated probability to all unlabeled examples. Next slide, please. For the KG2ML work where we used disease and genes, this is how we decided positive and unlabeled examples. For the positives, we selected all the genes which had link to disease through compound node. We call them positive genes, and all the genes which had link with positive genes through EFO nodes. We call them unlabeled genes. And the EFO nodes became our features. So now we have positive examples, unlabeled examples, and the features. Next slide, please. For testing the KG2ML pipeline, subject matter expert in our team suggested us these 12 diseases. And you can see we have a very significantly higher number of unlabeled genes and slightly lower number of positive genes for all these diseases. So these are the known genes which are associated with these given 12 diseases. And these genes have not yet been studied or there is no link in our DDKG. And for these 12 diseases, we found how many EFO nodes are there, which we used as features in our PU model. Next slide, please. So here you can see using our Pulsar method, we estimated what fraction of the unlabeled set H positive genes. So for these 12 diseases, the percentage is like higher. So what we did, okay, next slide, please. So what we did, we sorted all the unlabeled genes by their calibrated probability and selected top 15 and gave those top 15 to subject matter expert. He looked into the next database and literatures to determine whether the genes which don't have any link in the DDKG are really associated with those diseases or not. So you see a large number of those top 15 had yes. Yes means subject matter expert found association between gene and disease in the either TNEX or in some literature. And no means they did not find any association. So you can see a very large number of those top 15 have yes. So it means the Pulsar method can successfully identify some of the association between gene and disease, which are not present in the DDKG. Next slide, please. Yeah, another for example of those conditions we selected in our testing. Next slide, please. Okay, this is the recent work since in the previous work we used gene and disease. Recently, we tried our KG2ML pipeline to identify compounds which may be associated with a given disease. And we presented this poster at the CFDE spring meeting in Bethesda. So we selected two conditions for testing our KG2ML pipeline, type 2 diabetes and hypertension. And we use the same approach, we use gene as the intermediate node to identify positives and then unlabeled examples. And using pure learning method, we identify the top unlabeled or unassociated compounds and give those compounds to subject matter experts to determine whether those compounds are associated with the disease or not. And here, next slide, please. So amongst those top 15 for type 2 diabetes and hypertension, I selected two examples of folic acid and pterostilbene for type 2 diabetes and for hypertension, molsidumine and PCO4. And you see here, this compound is not directly associated with type 2 diabetes, but it is associated with type 2 diabetes through gene and some compounds which may treat type 2 diabetes. And similarly here, not directly associated with type 2 diabetes in the DDKG, but associated through these genes and some compounds which may treat type 2 diabetes. Similarly for this one, molsidumine and PCO4, again, is connected to this hypertension condition through some genes and compounds, and these compounds can treat hypertension. Next slide, please. Oh, I'm done. Thank you.
Thank you, Praveen. And My one comment, as you know, is we have these summer interns who have just begun their research, and some of them, I think two or three, are probably going to be working with the KG2ML approach. Some are quite qualified to perhaps even expand on the condensed knowledge graph. So we're excited about that as well. So just to wrap up, the take-home message that I hope you get is that the IDG program has produced a lot over its lifespan, over 500 publications, 79,000 citations, and continued productivity and increases their continued relevance and scientific impact. There are new, improved, and updated resources which are available for use and with opportunities for collaboration. Again, I like the analogy, whether it's good or bad, of aging gracefully, and IDG's in semi-retirement, but with some activities going strong and producing some new and updated resources. And perhaps the top news is the FAIR-ified 2.0 update that Jessica described, KG2ML work, which Praveen described, and the Drug Central update, which is in progress and expected later this year. So with that, I'll just finish off with acknowledgements, and it really is a large and extensive group of contributors and centers and institutions, US-wide and worldwide, who have really contributed to IDG over the years. And again, some of that is ongoing, and we're pretty excited about it. And I think that's it. Mackenzie, do I hand it back to you, or what do I say now?
Thank you so much for the presentation. Now we'll open it up for any question and answers. If you have any questions, please feel free to either use the chat, raise your hand, and we can take you off mute. Or if you want to use the question and answer option in Zoom, we can do that as well. So we have some time for any of those. So we'll wait a second and see what we have. Julie?
Yeah, I'll go ahead and kick us off. Thank you all. That was fantastic. I know, Jeremy and Christoph, I've met you guys, but haven't met the full team. We're big fans of the work that you're doing and excited to continue thinking about ways that we can bring IDG, further in with other CFDE resources as well. Jessica, I think you had mentioned during your part of the presentation some harmonization protocols that you guys had developed to make some of these ontologies BioLink compliant. So I'd imagine that some of these protocols are of high interest to other consortia that are looking to do similar things, right? To develop standardization across different resources and promote interoperability. Are those protocols available?
So I mean, everybody's doing it in silo. It seems like what because everybody has their own definition of reproducibility, and interoperability is what we're finding out. But since our group is embedded within NIH, we're trying to press a little bit harder. But that also comes to note that Translator is doing a giant step for that in itself. And so there's something called node normalization, which is I can never remember. It's the Renzi group, but so there's Translator itself has different people from different groups, just like the IDG. It takes the village, right? And there's these new versions that Translator is taking over, but essentially what's happening is the harmonization pipelines that were in our IFX, because here, bear with me, IFX is informatics core within the DPI department, which is, I can't remember now, but it's preclinical in informatics. And then that's in NCATS, and then NCATS is in NIH, right? So IFX, which is where the team I'm under, we have a lot of different databases and tools that we have contributed and developed and maintained. So FAIR-ified is being one of them. We have something called RAMP, which is metabolomics. We have CureID, which is rare disease patient, and then we also have GUARD, which is another rare disease initiative. And so all these different data sets, we are doing the base, like we're harmonizing that part right there. But that does include harmonizing open data sets. So like data harmonizing NCBI to HGNC to UniProt, like just for an example on the target level, right? So all these different open sources that we have, there needs to be still some harmonization. But like I said, Translator also does that too. And so aligning with them and seeing what the work that they've already done, utilizing that, and then harmonizing our data sets to that is what's going on. These pipelines that I'm speaking of, it's called Odin. It is public and it is on a public GitHub repo right now. But we're slowly doing it through the like a node-based level. So like we finished targets. Now we're working with diseases right now, and then we're going to move to drugs. But again, translators are high level. We're following translators pipelines first, and then we're harmonizing ours to that.
I'd like to just throw in a quick comment that, as Jessica pointed out, UniProt is and has been such a tightly integrated part of Pharos, and we've really appreciated all the work UniProt's done. Recently, we did meet with Kathy Wu and some of her team, and UniProt is doing their wonderful things. And so, they are both an interoperator and really a force for interoperability. So that, whether it's just the IDs or beyond that, that helps us, and I think they deserve to be acknowledged.
Yep, yep. So it's the same thing with the disease too. Mondo was an initiative like it depends like everybody's kind of drawing their own harmonization pipelines, like creating these ontologies, standardizations. And so it's still a big balloon and everything, and we're trying to like bring it in and tuck it in, and that's essentially what the translator initiative is about too. And that's a huge initiative with a lot of people involved. So IFX harmonization pipelines is a small small sliver of what that initiative is, but yeah. And if you go to the BioLink, I think I had the link in there. If you go to the BioLink data model, they have tools too that actually help you. You can it's scripts, and it helps you translate your data sets or your data tools or something into the BioLink data model as well. So they have a lot of different amazing tools that are already like available to the public to use.
Great, thank you. Any other questions? If you think of any questions also after this session, you could forward them to us and we'd be happy to share them with IDG as well. I'll give it another minute or so to see if we have anything else. I know sometimes it can be a little daunting to ask over the webinars, but we're happy to have any questions.
I guess one question for Praveen while we're waiting. Your work was fantastic. Thank you. I think this is a really exciting new direction that you all are taking with IDG, and I'm excited to see what you continue to do through some of your new work. I'm curious with the slide where you showed some of the diseases that you were examining, and the targets that came out of those diseases, and worked with subject matter experts to see if those checked out based on what was known. What's being done with the ones that did not have anything known by those subject matter experts, or didn't seem to match up with the known published literature? Do you have a sense of where some of those were coming from and whether those might be of interest to future investigation?
Yeah, I think that Jim, can you go to that slide?
Yes. That one?
Yeah. So here you see some of those genes, like if you can make it full screen. So although the Pulsar method assigned very high probability, but the subject matter expert, he did not find any association of these genes with the given disease in either TNEX or literature. So it may be possible that this gene has not been yet studied for that particular disease, like it's for bipolar disorder. And if in future someone decides to explore, this can be a new direction. Jamie, you want to add something?
Well, yeah, I mean, back when Jessica was a postdoc, she was in the wet lab doing the experiments to try to validate some of the, dark genome. Part of the answer, Julia, that's the point. When you have this understudied or unstudied genome, you don't know anything. And so then you need to actually, send someone, into the lab and try to, learn things. And that's actually what could lead to a new breakthrough in drug. So it's everyone wants validation, but the lack of validation is kind of an indication that why we're doing this.
That's what makes it exciting. Yeah.
Yeah.
Yeah, that's one of the reason why we selected positive and unlabeled learning instead of going with the supervised learning, where you need label positive and unlabeled, label positive and label negative. Since those genes which are not associated with given disease, we don't know whether they are positive or negative, because it may be possible that those genes have not yet been studied. But this approach gives us, instead of randomly selecting some genes and starting study, so pure learning method can try those genes, and scientists can start from the top ones.
Another bad analogy that you could think is that, maybe your financial advisor is saying to you sometime, how risky, do you want high risk investments or low risk? If there's a lot of knowledge, well, yeah, I mean, you might actually have something there, but, probably someone else is working on it. If it's, totally dark, that's high risk, but if it actually works, there's a big reward.
Yeah.
100%. So if folks are working in these disease areas, how can they find those sorts of targets, to pick out the ones that are sort of, most like understudied, but sort of top unlabeled genes?
Yes, so our Pulsar method, it assigns probability to all those unknown genes. So you can sort them by the predicted probability by the positive unlabeled method, and then you can select the top ones. That's the reason why we selected like top 15 and gave those genes to the subject matter expert to identify whether they are associated with a given disease or not. So you can just run the KG2ML pipeline and look into the predicted probability, sort them by probability, and then look into the top ones.
That's great. Thank you.
Yeah, thank you.
Awesome. We have a couple minutes left. So if there's any other questions, now would be your time to ask. All right. It seems like a little bit of a quiet crowd today, but I just want to say before we wrap up, thank you so much to the IDDG team today for joining us. We really greatly appreciate you taking the time to present. A reminder to everyone that our next session is going to be June 26th. So we'll see you in a month. So thank you so much again. We greatly appreciate it. Have a
Thank
great
you,
rest of
Mackenzie,
your day.
Yep.
Thank
Thank you
you.
for having me.
Bye-bye.
Bye.